Yeah, hello guys. I'll be talking about the tissue culture of tomatoes, and as you can see, we have different variety. This is tom one. We have tom two also, and we have tom three. All those varieties of tomato are gotten from the National Horticultural Research Institute, Ibadan, Nigeria. These are the three varieties of tomatoes we want to culture, and we have to do the surface sterilization for this culturing of tomato first. So we are going to use 3.5% sodium hypochlorite for the sterilization of these tomatoes. This is a standardized protocol which we have done already. So we'll be preparing 3.5%. 100 ml will not be enough for us, so we have to prepare 200 ml of 3.5% sodium hypochlorite. So we have to double it, which is 3.5 times 2, which is going to give us 7 ml of sodium hypochlorite. So we'll be measuring 7 ml of sodium hypochlorite and fill it up to 200 ml of distilled water. So we are using the 1000 microliter micro pipette. So 1000 microliter is equals to 1 ml. So we'll be measuring 7000 microliter, which will be equals to 7 ml of sodium hypochlorite. So we'll dissolve in 200 ml distilled water. So after measuring 7 ml of sodium hypochlorite, we are going to dissolve or fill it up to 200 ml, as you can see here. Just filling up the sodium hypochlorite with distilled water until it gets to 200 ml. Yeah, you can see 200 ml. So we can now label and pour into the Palmer bottle and label for easy identification. As you can see here, 3.5% sodium hypochlorite. So we have to pour the sodium hypochlorite into the different jars containing the seed, well labeled Hortitom 1, 2, and 3. So we are to sterilize, but before then we have to add a drop of tin 20. We don't have tin 20, so we are improvising with liquid soap. Just a drop of liquid soap in each of the Palmer bottle containing the different variety of the tomato seeds, as you can see. And then afterwards, we'll shake vigorously for 20 minutes. As you can see here, we are shaking for 20 minutes. We are supposed to use the orbital shaker, but because of the unavailability of light, we have to use our hands. Then after shaking for 20 minutes, we have to raise 5 times with distilled water. Raise 5 times with distilled water until there is no more foaming nature in the seeds. As you can see, we are raising here, discarding the water. We are raising here with 5. We're raising here with distilled water for 5 times. After that, you prepared your laminar flow hoods. Everything is intact. We have our sterilized media. Everything here is sterilized. The ties, the cutting wool, the instruments we are going to use. As you can see, these are the test tube containing the MS media. Everything is sterilized. Then placed under the laminar flow hood, which is also sterilized with 70% ethanol. After that, we on the UV light, which is going to sterilize the instruments and the media. Then now the works begin. We have to sterilize our hands first of all with 70% ethanol, and we have our scalpel, our forceps inside the soak 100% ethanol. Then you sterilize the surface of the ties with 100%, which is absolute ethanol. Sterilize the surface to avoid contamination with absolute ethanol. Then after that, you pick up the scalpel and the forceps, which is the surgical blade which has been soaked in absolute ethanol, and flame it on the Bunsen burner. Then allow it to cool for some time to avoid shock to the seed or the plant you're about to culture. So we allow the instrument to cool for some time. Then afterwards, we'll take our seeds, bring the seeds out from the bottle, place them on the ties, as you can see here, picking the seeds with the forceps. We are going to place 2 seeds per test tube containing the MS media. So we are placing 2 seeds per test tube containing the MS media. Those seeds are very, very minute, so we have to be careful while placing them. First of all, We'll flame the tip of the test tube and the cover to avoid unnecessary contamination. This is the MS media inside the test tube. Then we'll place 2 seeds inside the MS media. As I said before, those seeds are minute, so we have to be 100% careful while placing them. Make sure that the seeds is on top of the media. It's placed directly on top of the media, as you can see. She's ensuring that the seeds are placed directly on top of the media. So this is the process was repeated till we had everything completed. As you can see, we have 2 seeds per media inside the test tube. Then taken to the goat room for 10 to 12 days before we subculture for